show you how to post that on YouTube later. Yeah. Anything else you guys would like?
Do you have any cookies?
Yeah, I do. Okay, next. You know, one of the things I'll show you from this first slide, of course, it says here that fertilization is, which is when the egg and the sperm meet up. It says here that's when development begins, and that's certainly true. There are many people in many different religions in the world believe that that is when life begins. But as I'm going to tell you later, that's true, but it's only about 60-40 because most fertilized eggs don't make it. And most of them just don't make it, period. Anyway, embryological development, this is a word that you ought to know. Embryology is basically the first 2 months after fertilization, because during those first 2 months is when all of the organs basically form. After 9 weeks, which is, you know, not even a quarter of the way through pregnancy, we call it a fetus because it pretty much looks like a little person in there. So from 9 weeks to 40 weeks, the growth of the fetus continues, but most of the basic development has been done by then. And the point I would make about that is if all of the development occurs in the first 9 weeks, and since some of you ladies don't even know you're pregnant until you're 5 or 6 weeks pregnant, you know, if there was going to be any problem with anything you were drinking, eating, or smoking, you probably should have not been doing it. You know, it would be too late to stop then. And the fetus would have suffered the effects of those things. So that's my lesson as your father. Prenatal development, we're going to go through that. Postnatal development, that just means post-birth. And obviously that continues at birth, and it continues until you die. Through that period of time, you develop and you age, and we'll discuss those things. Inheritance and genetics are basically the transfer of traits from parent to child. And inheritance is more of a loose term. Genetics is how it actually happens with DNA and all of that good stuff. Fertilization, I've mentioned this a number of times. Basically, it's the fusion of 2 haploid gametes. Remember, haploid means half, and in humans that is 23. So 46 is what every cell in our body has. Every cell in our body has 46 chromosomes. That means there are 23 pairs. You know, because of the wonders of meiosis, that cuts us down from 46 to 23 in sperm, and the same thing in oogenesis, and eggs only have 23, so that when they come together, they make 46 again. And we'll talk about every step of that. Spermatozoans, they are spermatozoons, I guess. They're the thing that they're going to bring the paternal chromosomes to the fertilization. They travel a pretty long ways. They're pretty small, they're pretty efficient, and they're highly streamlined because they've got to move on. The gamete is when the egg and the sperm come together and form a fertilized egg, and we call that a gamete. And of course, the egg mostly provides most of the other stuff, including the nourishment comes from the female side of things. The fetus floating down the fallopian tube gets no help from anybody for about a week. It's just kind of floating free there. The sperm goes up and meets it. The egg finishes meiosis, the last 2 steps of meiosis. It becomes a fertilized egg, and then it's just kind of bouncing on down the oviduct and into the uterus and trying to implant, getting ready to start implanting. But for that first week, it doesn't get much help. It's pretty much out there on its own. Fertilization usually occurs within about a day after ovulation. Oocyte doesn't travel very far. The sperm must travel all the distance between the vagina and the ampulla, because that's where fertilization generally occurs. Capacitation, I talked about during male development. It's kind of the final activation step of the sperm that turns it on. It has to not only contact the secretion of the seminal glands. Now remember, that's kind of a generic term there, because there are 3 glands that make semen. You remember, there's the prostate, there's the seminal vesicles or seminal gland, and there's the bulbourethral gland. So those 3 glands make 99% of the semen. The only 1% is the sperm that are floating in it, and then those sperm cells have to be exposed to the conditions in the female reproductive tract. In other words, it has to be kind of acid, a little bit of acidity inside the female reproductive system in order to really finally capacitate it and make the tail start to flip and make it really go. Hyaluronidase is this enzyme that's found between the follicle cells. Remember, even after it's ovulated, you still have this situation where there's an egg that is still surrounded by a bunch of follicular cells, so that it even after it's coming into the uterus, it still kind of looks like this with all these follicular cells around it. And when the sperm arrives, it has to find a way, the little guy has to find a way to get through those cells and get at the actual egg itself. So hyaluronidase is an enzyme that helps break down these bonds between the follicle cells and leaves room in between so that a sperm can actually get to the wall of the egg, illustrated by this big circle edger here. Acrosin is that enzyme that's right in the head of the sperm, remember, in the acrosomal cap. It's also a proteolytic enzyme, which means it eats through protein, and it's got to use that to help get through these guys, but also then burrow a hole through the wall of the membrane here to actually get inside, because the sperm has to get all the way inside to do its thing. I think kind of said this, acrosomal cap has these 2 enzymes in it. They've got to go through the corona radiata, the zona pellucida, before that's these several layers of cells around here in order to get to the oocyte surface itself. Usually, once the fertilization occurs, once the sperm actually makes contact with the fusion and fuses with the cell membrane, then it allows the DNA from the sperm to get inside. That also signals to the egg, to the oocyte here, to go on and finish up with meiosis 2. Remember, because until that point, it just was still hung up at what, meiosis 2, during right before metaphase, isn't it? Anyway, y'all know. I don't. Once the male DNA has gotten inside, we call that the male pronucleus. The female pronucleus is the female's chromosomes that are already in the egg. So the bottom line here in fertilization is that the female pronucleus and the male pronucleus are now going to both be here in this oocyte, and they've got to get together. And so here's the female pronucleus over here with its DNA. This has got its DNA. The tail's going to go away. And these 2 things have got to then hook up together, and they've got to pair up. They've got to pair the chromosomes together. So chromosome 1 has to hook up with chromosome 1 from the daddy, and from chromosome 1 from the mommy. And chromosome 2 has to line up with chromosome 2 of the daddy, back and forth until there's a whole set. So now we've got a new set in the middle of the cell. That is called amphimixis. Kind of interesting. interesting word. And amphimixis is really the fusion of the male and the female pronucleuses. So when these two guys actually hook up together, that can be called the moment of conception, because that's when their DNA actually hooks up together. And at that point, the cell becomes a zygote with 46 chromosomes, and fertilization is complete. It's still just one cell, though, right? One cell with 46 chromosomes. And then things have to start dividing, and that's called cleavage. There's a series of cleavages that occur, and one cell goes to 2 and 2 go to 4. Each of these is identical in every way. It's just simply mitosis that's going on, and you're just creating daughter cells that are exactly the same as that original one cell. But at some point, they're going to have to start differentiating. This is a picture, a micro picture of a poor little egg, poor big egg being surrounded by lots and lots of little sperm cells trying to get into it. Only one is going to get in, because once that one gets in, then it just kind of shuts down and won't let anything else in. Here's the oocyte ovulation. It's still got to do that last division and get rid of that polar body. Let's see, there's the first polar body. It's going to divide here and create the second one. Here, now you have the second polar body created. This is really schematic that these two guys are lined up here together, because I don't think it really happens quite that way. But this is the female pronucleus right here. This is the male pronucleus coming there and coming into the egg cell. They line up here, male and female together. Sperm is absorbed. The tail is going to disappear, and then they're going to line up in do-si-do, so to speak, as illustrated right here. So this is where they all line up and they all pull apart, as illustrated here. Now these are exact copies of each other. So you're splitting all 46 of them and getting identical twins, basically, for children's cells over here, called blastomeres. The first cleavage division doesn't even happen for about 30 hours after fertilization. So once again, a very, very critical time in life is that first 30 hours when this thing tries to split the first time. First trimester, second trimester, third trimester, you guys have all heard those terms. Basically, they're about 12 weeks apiece. That's kind of a rough number, because 12 times 3 is only 36. Normal pregnancy is 40 weeks. We call that the gestation period. And for humans, the normal gestation is 40 weeks. As I said before, in the first trimester, almost all of the major organ systems appear. If they haven't totally appeared, at least they're rudimentarily there. Second trimester is really when body shape and proportions change. And by the end of the second trimester, it really does look like a kid, a baby. All that happens in the third trimester is everything just gets bigger and bigger and bigger. And that's why the third trimester is so important. If the baby was born at 28 weeks of development, it would weigh about 1.5 pounds. And yeah, babies can live these days at 1.5 pounds. But, you're talking $500,000, because you're talking 12 weeks in the neonatal ICU. And it's $10,000 a day or more to keep those babies going. So that's why the March of Dimes and other organizations work so hard on trying to help ladies with their pregnancies and keep them in there, because there's no better oven for them to keep cooking in than the mama's uterus, and that's a lot cheaper to run than an incubator, which is very, very expensive with all the nursing manpower and doctor manpower that's required to make that thing work. Cleavage does not refer to the breasts. It simply refers to splitting of the cells, and it just starts to divide over and over and over again until you get basically a big ball of cells. And that's what we call the blastocyst. There's a few words along in here that you need to know. I think if you read through your book, they'll come to you very easily. The blastocyst. You see, this has all been dividing as it's still bouncing down the fallopian tube, and usually the blastocyst is eventually going to touch up against the uterine wall, and it's going to start eroding into the uterine wall in order to implant, and so it can steal, oxygen and sugar and all that stuff from the mom. Implantation is the name given to the attachment of the blastocyst to the endometrium. And you remember from our talk about the uterine cycle that the uterus is working really hard to get ready for that fertilized egg to come down there and implant in it. And so remember the secretory endometrium gets thicker and thicker. All those glands grow. They make lots of healthy wetness on the inside of the uterus, ready to receive that embryonic structure. Another funky word is placentation. Placentation just means formation of a placenta. And anybody know what a placenta is?
Isn't it an organ that's only with you while you're pregnant, and then you get rid of it? Is that what you mean?
Yeah, it is. It's something you get rid of with delivery.
It's almost like a phospholipid bilayer for a fetus, in a way. Protects it from getting in.
Well, let me just say that having delivered 33 placentas in my life, the placentas are about this big around. It's about that thick, and you probably know of it as the afterbirth, because it generally, if you've ever seen an animal deliver, the baby comes out first, attached to the umbilical cord. That umbilical cord is still attached to the placenta. But the placenta is this growing thing that attaches to the wall of the inside of the uterus and kind of sucks the blood and so forth. The placenta has two sides to it. It has the maternal blood on this side. It has the baby blood on the other side. And it's where all the exchange takes place. So it's a great big membrane with baby blood on one side and mama blood on the other. But it's very important that the placenta become very healthy, because the placenta is the thing that's going to be responsible for getting all the nutrition for the baby. And if you remember me talking about ectopic pregnancies, the problem with ectopic pregnancies is, a little placenta can start to form, and it can maybe get that big on the inside wall of the oviduct, or on the inside of the duct, but it needs to get this big to support a fully grown fetus. So that's why they don't really work out. And ectopic pregnancies almost never make it to term. So anyway, placentation is this formation of that placenta. Remember that the placenta is just a whole bunch of blood vessels, and they're both maternal and baby. The placenta immediately, once the baby comes out at delivery, the baby comes out and the uterus goes from this big down to about this big, right? Because it's just pushed the baby out, so he gets a lot smaller. So that big old thing that was this big around, that was hooking onto the wall of the inside of the uterus, is now basically, because it's all shrunk up, it basically pushes the placenta off. And the placenta separates from the inner lining of the uterus, and then the placenta is passed out. And that's one of the biggest problems in delivery from a life or death point of view, is it's very important that the placenta does completely separate. You don't want to leave a part of the placenta there, because that piece of placenta will continue to bleed because it's nothing but a bunch of blood vessels, so you kind of got to get all of it out. And sometimes we don't. And that's why some ladies end up going back to the operating room the next day and having a little mild DNC to scrape any last placenta that's left in there out. But the main thing that causes it to separate is simply the physical fact that the uterus goes from this big to, say, this big when the baby is delivered. And so that usually causes the placenta to start breaking off of the inner lining of the uterus, and then hopefully within about 10 or 15 minutes, it gets ejected out and delivered following the baby, and it's called the afterbirth, as I mentioned before. Embryogenesis, I think we've mentioned that. The most dangerous period in prenatal life is in the first trimester. Only about 60% of embryos make it through the first trimester. In other words, an awful lot of pregnancies are lost. A lot of times the lady doesn't hardly even know she was pregnant. A lot of times she does. And if I could give you another word of fatherly advice, is if you get pregnant, keep your damn mouth shut about it. Talk it over with your partner, whoever co-sponsored this new living thing, but don't tell anybody else until you get to be 10 or 12 weeks. Everybody wants, oh, I missed my period's a week late. Oh, Mom, I'm going to have a baby. Don't do that, because all you can do is break your mother's heart if you lose the pregnancy. And lots of them are lost early on. So my advice to people, for what it's worth, is just keep your mouth shut. Keep it between you and your partner. And as it gets a little bit farther along and the odds become a whole lot better that it's going to survive, then tell everybody and be happy. For what it's worth. Okay, blastomeres are these big clusters of these cells. They just keep dividing, and they're all the same. They're produced by a whole bunch of cleavage divisions. And then after a bit, we call it a morula. This is really a silly name. It's called a morula for no reason other than the fact that it looks like a mulberry. A mulberry looks like a raspberry, I guess. It's just a whole bunch of little, it's a ball of a cluster of cells. This morula is found in about 3 days, and it's probably 100 to 250 or so cells in a cluster called the morula or mulberry, and it's still rolling on down the duct towards the uterus. Then it becomes called a blastocyst, and then a blastocele, and all that amounts to is that these the blasto thing starts picking up fluid inside of it, so it kind of becomes a hollow ball, and all of the cells are around the outside of it, and the inner cavity is known as the blastocele. The opening, the lumen is called the blastocele. Another thing that starts to happen about this point is now you've got this ball of cells that is this morula, and it's come up against the wall of the uterus like that, and it starts eroding into the wall of the uterus. And that's a good thing. Obviously, it has to happen, but sometimes it goes a little too far. And this trophoblast, which is the part of the blastocyst that is actually going to become the placenta as it starts to erode into the wall of the uterus. So let's go a little bit farther and get to this thing called the inner cell mass. And basically what happens there is you've got, if this is your blastocyst, my hand here, and it comes up against the wall of the uterus, then it's going to finally start to develop, instead of being just a round ball of cells, it's going to develop a right and left, it's going to develop a top and bottom, it's going to develop a head and tail, so to speak. And the part that erodes in is called the trophoblast. The other area, which is kind of on the back side of it, which is called the inner cell mass, is at the other end of the blastocyst, and it is the part that is going to become the baby. the inner cell mass is going to become the fetus. Okay? I got pictures that will help explain this better. But here you go. So here's your first cleavage division. 1 becomes 2, becomes 4, becomes 8, becomes 150 or so, and we call it a morula, this mulberry thing. And then here comes this mulberry thing, and this mulberry thing is right over here now, and it's gonna move over against the wall of the uterus, and notice that it's gonna start eroding right into the uterine wall. And that's called implantation, which you see right here. And then you have 2 ends to it, and this end right here is called the inner cell mass. And I think for your understanding at this point, all you need to know is the inner cell mass is what is going to become the fetus. Okay. So this is 7 days down the road. This is 7 days since fertilization. This is probably 9 days since sex, and the spermie's starting to swim. And now we've got our blastocysts adhering to the uterine lining. And these trophoblastic cells, the trophoblastic cells are the ones that are eroding into the uterus and creating a placenta. Okay? Here's a nice set of small pictures of it. Here's your nice, thick, juicy, lots of secretory cells in the wall of the uterus, the functional zone of the endometrium. This blastocyst starts moving over against it. This is the inner cell mass right here. This backside of it's actually going to come the trophoblast. And you notice that 2 things happen. First of all, the mama starts developing a syncytial trophoblast, which is going to be sort of her contribution to this placenta thing. And the baby is creating the cellular trophoblast, which is kind of going to become the baby's side of things. And you're noticing that this whole apparatus here is kind of getting big, and that's what happens. Little lacunae form. Lacunae are nothing but pools of blood. This is the mother's blood being poured into here, which is what's going to nourish the baby through here. I've talked about ectopic pregnancies enough. I don't think I need to say anymore, but just remember that the lacunae are these trophoblastic channels that are carrying mama's blood. Villi start to extend deep away from the trophoblast into the endometrium, and they keep getting bigger and bigger until about day 21. Now let's go back real quick and look at that. This is the trophoblast growing in here into the uterus wall. Lacunae are forming. This is mama's contribution to the placenta. The amniotic cavity is the fluid that the baby's going to be floating around in eventually, and the inner cell mass becomes organized into an oval sheet. Now here's where it really gets amazing to me, because we started out with a big ball of cells, remember? And then the cells kind of moved to one side and became called the inner cell mass. And now those cells flatten out, and we have a whole plate of 2 cell layers thick on the outside, what is going to become the outside, or the ectoderm of the baby. Ectoderm skin, okay? Ectoderm is also going to become brain, okay? But that's all just a flat sheet at this early stage. And the opposite side is going to become the endoderm, which is going to become the inner lining of the GI tract from mouth to anus. Okay? That's really not in you, remember? I've said that from the beginning. But that's going to be the endoderm, which lines the inside of the body cavity, basically the inside of the GI tract. We get to this point now, which we call gastrulation. I think it's kind of a silly name, but gastrulation means that the fetus is going to form a 3rd layer of cells. So up to this point, We had the ectoderm, which is going to become the skin on the brain. We had the endoderm, which has become the inside lining of the GI tract. But now we're going to develop a third layer. And the third layer is going to be called the mesoderm. And it's going to be everything else. It's going to be the bones, the muscles, all the stuff in between. All right. In order to make this thing work, you develop this streak and this ridge. So I told you it was it was 2 cell layers thick. And if this is the big plate of cells, it kind of looks like this. But then it kind of starts developing what's called the primitive streak. And so it develops 2 kind of thicker layers here of ectoderm, of skin and brain, right? And one of them becomes the head, one of them becomes the tail, and this ectoderm, the outside, is going to kind of fold up and over and form the brain. And the stuff in the middle, down the middle of that ectoderm, is going to become the water that runs down through the middle of your brain, which we call the CSF. And the water is in little cavities we call ventricles. And it's also in what's called the center streak that goes all the way down the spinal cord, which is called the central canal. Right? Remember that guy? Okay. So the brain, the ectoderm, has to come up and form around this and seal together over the top. Okay? We'll talk about that. Because if that doesn't happen right, it leads to bad problems. So here's the ectoderm. Endoderm is the cells that face the inside, and the mesoderm starts to form this middle layer of cells. Looks like this in your book and looks like this in real life. So that here's the ectoderm. This is the blue outside. It's going to become your skin, and it's going to become your brain. Okay? This is the streak down the middle. These 2 things are going to flip up and over here to enclose the brain tissue. This is the endoderm down here, which is going to become the inner lining of the GI tract. And then in between those 2, it starts to form this third layer of cells, which are illustrated in red here. And those red cells are going to come the mesoderm, and what they're going to become is the blood, the heart, the blood vessels, the muscles, and the bones. Everything that's sort of in between is going to become the mesoderm. It's going to be made up of the mesoderm, as illustrated right here. The bottom part, this is going back to this one here, for it's quite as advanced as that. And this becomes the yolk sac down here. You guys have all cracked eggs. And the yolk sac, which we humans have too, it's not in an egg or anything. It's just a structure here. And you notice that it's lined by the endoderm, which is going to be kind of incorporate this into the GI tract. And it's going to become food, and the first round of food in the GI tract is really from the yolk sac. Here's this disc. We call it a disc once it gets to have 3 layers. The 3 layers are the ectoderm, the mesoderm, and the endoderm. Most of it's going to become the body of the embryo. The rest of it is just going to become these embryonic membranes, which I don't really hold you to. I don't want you to learn all the membranes because they're just awfully complicated, and I don't think it adds a lot to your understanding. And here's the list of them, and you're certainly welcome to read through the parts in the chapter and make yourself understand these. But the extra embryonic membranes, that means they're outside of the embryo. They're extra embryonic. They consist of the yolk sac, which I just mentioned, the amnion, remember amniocentesis, baby floats around in amniotic fluid, the allantois, and the chorion. They're all membranes that kind of go away, except for the amnion, because at birth, the baby is still floating in the amniotic sac and in the amniotic cavity. So it starts looking kind of like this, and now you can really start seeing the three layers. The blue is the ectoderm, which is going to become the brain, remember, as it folds over. The middle part is in here is the body, basically the muscles, the bones, blood vessels, heart, all that stuff. And then the endoderm, which is the inner lining of the GI tract, also touches up against the yolk sac, which is illustrated right here. Now, you can probably guess from looking at this right here that this is the placenta. It looks like it's all the way around, but it's gonna retract and just be on one side and be that disc that I've been telling you about that big. And then this is going to become the umbilical cord. Okay. Let's go a little bit farther along, and it looks kind of like this. Now we can say this is definitely going to be the head end. This is definitely going to be the tail end. Once you got a head and a tail, then you've got a right and left. Sounds kind of silly, but the fact is you've got directions that are going to happen. Notice the yolk sac is being kind of sucked into the endoderm in here, and it's being eaten up. Yolk sac usually disappears completely, as you can see it going on here. Here's the umbilical stalk starting to form, and that placenta that we looked at a slide or two ago, remember, and it went all the way around. The reason it went all the way around because this whole thing was in the wall of the uterus. But as it gets a little bit bigger, then the placenta localizes itself to one side, which is up against the uterine wall, and the rest of this just fills in all the rest of the space as mama gets bigger. Does that make some sense? Here we go. We're up to week 10. We're almost developed, really. And now you can see the fact that we have the placenta is all on one side, which is where we want it. One place we don't want the placenta to form is down here, because if it formed over the opening of the cervix, that would be a bad deal. The baby couldn't get out through there, and that is called a placenta previa. And the only way to get that baby out alive is to take it out to C-section. And that's one thing ultrasounds are really good at, is they can localize where your placenta is. So now we've got a placenta formed on this side. You notice that there's mama's blood is out here in these lacunae things. This is baby's blood is collecting and going up through the umbilical arteries, right? And it's coming back through the umbilical vein, going back to the mama's circulation, and we'll get into that a little bit further. Umbilical cord is just the thing that connects the fetus to the placenta. It contains the allantois, the placental blood vessels, which is 2 arteries in one vein, and the yolk stalk. Blood flow, remember from your blood vessels back in 232, the blood vessels to the placenta come off of the iliac arteries down in the pelvis. And remember, they bend up and they form those umbilical those vesicular bladder, the bladder ligaments, the lateral bladder ligaments, and they come up to the belly button, and then they go out to the baby, and then they come back through the vein. From the vein, they then go up, go through the falciform ligament, go through the, what's that bypass called? Ductus venosus, right. Put all your heads together and you'll get it. Yeah, the ductus venosus, which carries the blood directly from the belly button, basically from the umbilicus, through the liver, straight into the inferior vena cava, where it goes on back to the baby's heart. Now you're starting to see this develop a little bit further, and so this is maternal blood out here in these big lakes, and the baby has created, the placenta has created these villi that extend out into that mama's blood, and this has both arteries going to the baby and veins coming back that are carrying the oxygen and so forth that is being picked up in the placenta right there. This is what it looks like under the microscope. This is one of those chorionic villi, which has lots of fetal blood vessels in it, lots of embryonic connective tissue. These are just sticking down into those lacunae of mama's blood in the placenta. You can imagine how much the placenta would bleed if it ever got torn or hurt. And just so you know, the standard blood loss in a perfectly normal delivery is over 1 liter of blood. That's normal, totally normal. This is a bloody organ. Okay, what does the placenta do? Well, it secretes a lot of hormones, and the biggest one it secretes is this one here, HCG. Anybody know what HCG is? Well, it's human chorionic gonadotropin. What do we use it for? We test for it. And what do we test for?
The numbers to rise.
We expect the numbers when the minute you get pregnant, and once the implantation starts to happen, then HCG is released from the early stages of the placenta out into the circulation. And that is your first pregnancy test. Now, HCG, so it can be detected in all these little things you pee on, and a little plus shows up. All they're testing for is this right here, HCG. Now, as you go through your pregnancy, if you were to lose your baby in the womb, the HCG levels would crash back down, and that's another piece of information, because sometimes the diagnosis of fetal demise is not easy to make, especially if it occurs before movement is detected by the mother. You know, you can take an ultrasound and look for the heart beating and all that other stuff, but HCG is used in that as well. I had an OBGYN guy in Dallas. He said, I never order a pregnancy test. I said, really? He said, yeah, I never do. I said, I'll figure it out. They were either going to be pregnant or they're not, and who needs a urine test or a blood test to figure it out? And I think he was making a good point, because every lady in the world wants to run into the doctor and get a pregnancy test, you know, right away. He would always say, no, let's just wait a week. We'll know. And maybe it makes kind of sense. Anyway, these other hormones, I don't think you need to worry much about them at this stage. Relaxin is a hormone that's released that allows women's bodies to really kind of soften up, allows their pubic symphysis eventually to open up to make more room for the baby to get out. Progesterone, of course, supports the placenta and the pregnancy. Estrogen, female hormones, they're making all this thing work too. HCG is a very reliable indication of pregnancy, and if HCG is gone, there is no pregnancy. That's just the way it works. HCG also helps prepare the mammary glands for milk production after delivery, and serves as a stimulation to other tissues down there to make them all grow, kind of like growth hormone, making the placenta grow, making the uterus grow. The uterus is going to have to grow, remember, from the size of 2 thumbs into this kind of massive, many, many pounds of muscle that holds a full-term baby, and then has to push it out. Relaxin is secreted by the placenta and the corpus luteum. It makes the pubic symphysis more flexible, allows the pelvis, one of those 2 big joints in the back of the pelvis called the iliosacral joints, you know, where the ilium and the sacrum join, causes the cervix to start to dilate down here to allow the baby to get out, and it it suppresses the release of oxytocin. Now, oxytocin is the hormone released by the hypothalamus, which is actually going to cause the uterus to go into labor and keep the labor going. So at first, you want to continue to suppress the oxytocin. You don't want the lady to go into labor early, right? That would be a bad deal. Embryogenesis, yeah.
I do have a question. So, the relaxin says dilation of the cervix, but suppresses the release of oxytocin. When your cervix is dilating, you're begin labor. So then,
That's right at the very end. Yes.
So once the cervix starts dilating, the relaxin turns off and starts releasing the oxytocin.
It allows oxytocin is remember negative feedback and positive feedback. Oxytocin is like one of the ultimate positive feedbackers. Once you get a little bit of it, you release more and more and more, and you go and labor becomes more and more sustained and more powerful.
So I was just kind of wondering if there's like a relationship between the two. So if you start dilating, then you start release oxytocin, or are they just independent?
I can't answer that question. I would say they're both working independently to some degree.
Okay.
We've been through this. I don't think I need to say anything more here. I told you when I was drawing this picture over here, and I drew that streak, and I said that the ectoderm built up on two sides of it. This is what that looks like early in the first trimester. This is the head of the fetus. This is what the lower end looks like. This is where the tail would be. So this is the axis of that spinal cord, and these parts of the brain here are going to come over here and unite together in order to encircle the ventricles and the fluid that runs down the middle of the brain. I'll talk to you about what that means if that doesn't happen a little later.
Oh, I think so.
Yeah, spina bifida is one of the things. So here's a nice photograph from week 4. Notice that the forebrain has moved and fused around. Notice that the pharyngeal arches, that's these guys right here that in evolution, if you've ever studied evolution in detail, the arches that we have in our neck are exactly equivalent to the gills that are found in fish. We developed lungs instead to collect our oxygen from, but so that's what the pharyngeal arches are right through here. And then notice as you come on down here, you have these things called somites. Somites just means segments of the body. And you think about this vertebra, the vertebral column, each one of those is a somite. And it's going to send nerves that nourish the muscles that are in that somite that make the muscles in that section work. Here's the leg bud down here at the bottom. There's the heart, 2 chambers. Doesn't look like, if this was moving, I'm sure there would be some, at least twitching of that area in terms of early heartbeat. Here we go a little bit farther along. This is 8 weeks. Here's the amniotic cavity right here, umbilical cord, your chorionic villi of the placenta out there. Here's another baby photograph. This is a pregnancy loss, I can tell you, because obviously it's out, and the whole amnion is surrounding this fetus. And you notice the fingers are starting to look like fingers a little bit. This is week 12, and as I said, at the end of the first trimester, the baby really does sort of start to look like a baby. Second trimester, everything's growing. Third trimester, everything's getting bigger. The fetal heart rate when it started was well over 200. It's really, really fast. But then as it gets closer to delivery, the heart rate's down to like 150. And it's still much higher than our heart rates, but quite high. Mama starts feeling pregnant. All of her organs get pushed around. Baby seen through a fiber optic endoscope there. Isn't that cute? I would just say that the pregnancy at 4 months is usually pretty invisible to most anybody. But once you get beyond 4 months, then the top of the uterus starts growing like this. And then just before delivery, when you get to 9 months or so, everything drops because the baby's head moves all the way down to the bottom of the pelvis, and that also starts helping to open up the cervix to make room for the baby to come out. So it's not unusual, they say at 36 weeks for the lady to be 3 centimeters dilated. I mean, the dilation can, when I say dilate, I mean the cervix, the mouth of the cervix is starting to open up. Here's what it does inside of you, ladies. Here, the head hasn't dropped yet in this picture here, but notice that everything else, all the guts and everything are pushed way up into your throat. And the bladder, poor bladder down here, gets smashed by the baby's head. So if you've been through a pregnancy, you know you kind of had to check in with the bathroom about every 20 minutes usually, because there was just no room to store urine down there. False labor is what happens towards the end of pregnancy. Often it's just an occasional spasm. The only thing that separates false labor from true labor is the fact that true labor is really caused by the hormones and the oxytocin and so forth. And it is a labor that begins and then it gradually gets stronger and it actually gets closer together so that the contractions become much more regular. And when they get down below 2 and 3 minutes, you know, you got to get to the hospital if that's where you're going to deliver your baby. So the labor contractions, I've helped a bunch of ladies get through labor and have babies, and I think one of the most amazing things to a male observer is to watch the uterus contract, because it's this soft thing, you know, you can kind of push around on it. It's not a big deal. But when she's having a labor contraction, that uterus just kind of stands up and it gets absolutely as hard as a rock. And I mean, I'm not exaggerating. It is hard as a rock there in the abdomen, and you can see it from across the room as it's just like, man, that thing's alive. And it is. And as the labor contractions continue, then it continues to open and push the baby on down. Parturition is another fancy name for birth. I think I don't need to say anything. Dilation is simply the opening up of the cervix. If you ever work in OB, you'll learn how with your fingers doing a vaginal examination to tell how many centimeters the cervix is open. It's really easy if you can put one finger in it, and you can feel it easy enough. If you can put one finger in it, then it's about 1 centimeter. And then when you get up to 2 and 3 centimeters, it's 2 or 3 fingers, and then you get to the point when it's 7 or 8, and you're actually moving your fingers back and forth across from one side of the cervix to the other as it opens on up. Labor usually lasts 8 hours or so. At some point in there, the water breaks. I remember Joan Rivers used to always say that she got to be past about 7 months of pregnancy. Every time she went to the grocery store, the first thing she would do was go to the pickle aisle and grab a big bottle of pickles and put it in her shopping cart, so that as she was going around through the grocery store, her water broke, she would just take and drop the bottle of pickles and say, clean up on aisle 6. And she didn't have to admit that it was her. But anyway, she told it a lot funnier than I did. But anyway, so here's birth. Birth kind of works like this. Usually the babies come out sideways. They're facing one side or the other. They almost always come out head first. As they come out, they rotate so their face is down and back. When they come out with their face up, we called that sunny side up, and that was pretty rare, and it was usually a lot harder to deliver the baby sunny side up than if their face was down, just because of the way their neck bent as they made this big turn right here. Expulsion stage is the last bit. This is the dangerous part of labor, because this is when the baby is usually oftentimes during the second stage of labor here is when they oftentimes will have slow heart rates, and their heart will go down on every contraction. Heart rate will go way down sometimes, and those are times when brain injuries can occur, And so that's why most babies are monitored during these last few stages of delivery. And if their heart rate is dropping too low, they're usually going to get a C-section, or something is going to be done really quick to get that baby out of there, because you don't want to leave it in that situation for too long. Here it comes. It's got hair. The placental stage is once the baby is born, then you got to wait on the placenta to be born. And there's always a loss of blood because that placenta is ripping itself loose from the inside wall of the uterus. And so here, and the uterus has obviously shrunk up a lot, and as it shrunk up, it kind of pushed the placenta here off the inner lining. And as the uterus continues to contract, the uterus squeezes off all those massive blood vessels that have been running through the wall of the uterus to nourish that baby, and that's what stops the bleeding. Usually once the baby is out, once the placenta is out, the patient is given a bunch of oxytocin in the bag. Pitressin is the trade name for it. And that causes the uterus to really squeeze down hard, which is what you want to have happen so it doesn't continue to bleed from where it's just pushed off the placenta. Episiotomy is an incision made through the wall of the vagina and through the muscles around the vagina in order to make room to let the baby come out. I don't think this is done near as often as it used to be. We used to do it on every single primip. In other words, every first child, they were going to get an episiotomy. Basically, you put your fingers into the vagina. The uterus is, the skin is just pulled tight as a rubber band around there. Put your fingers in there, put one blade of the scissors inside the vagina, one on the outside, and just go boom, make a cut about that long. And that opened it up enough for the baby to come out. Now that sounds pretty brutal. And what's happened is that women have said, wait a minute, we don't want to have those incisions made down there because then you got to sew them up, right? And so now there's all kinds of stretching and oiling and greasing and continual dilation all through labor, trying to stretch the vaginal wall out so that that's not necessary. Now, quick story, episiotomies were done so that when you made a cut, you cut through all the muscles down there, and it went out to the side. And when it's cut straight, you can sew it back straight. And, sewing up an episiotomy took, 10 or 12 minutes. It was not that big a deal. It was all disposable sutures, and they went away, and I never saw one of them really get infected. So from my student position as a medical student, it seemed like the right thing to do. Women don't like to have that done, but just so you know, I have a sister-in-law who was delivered with the best of gynecologic care. She went and started seeing the doctor when she was a month pregnant. She went all through her pregnancy. Everything was perfectly healthy. She got everything was done right. She got to the end, and the doctors decided not to do an episiotomy, and she bared down that last time, and everything ripped, and out comes the baby, and the baby's born, and the baby's fine, but she had a rip all the way down into her uterus, all the way into her rectum. It tore through all of her rectal muscles, it tore through her bladder muscles, it tore her vaginal wall to shreds. And because it was ripped like that, you can't really sew it back together very well. You try, you get really good lights, and you use little bitty sutures and try to figure it out, but that's the reason for episiotomies. Episiotomies give you a nice straight cut where you can put the two sides back together a few minutes later, whereas a fourth-degree vaginal laceration is a terrible thing. She was incontinent for over a year before she could control her bowel movements. That's not a lot of fun when you got a baby to take care of. So I just mentioned that. Excuse me. C-sections is taking the baby out through the tummy up here. It can be done lots of ways, but basically the incision is made right at the top of the pubic hair usually, and a little incision about that long, and then you go in there and make a transverse incision across the neck of the uterus, and you open the uterus up just enough to be able to pull the baby's head out. Once you get the baby's head out, the baby lifts out of the hole pretty easily. Put a clamp on the umbilical cord, cut the umbilical cord, pass the baby off. Breech birth is feet first. That's a problem. Let's talk for just a minute about twins. Most twins are dizygotic twins, which means there's two zygotes. Some ladies, when they ovulate, instead of ovulating one egg, they'll ovulate two, and both of them get impregnated by a separate sperm. So basically, it's just like having either brothers or sisters. They just happen to be living in the uterus at the same time for the same 9 months. This develops whenever there's two separate oocytes or ovulated. So they are not identical twins, and indeed about 7% of twins are this kind of twins. And as I said, they're nothing more than brother and sister. Identical twins result from one of two things. Either at the early stages of the blastomere, as those things are dividing and dividing, they separate from each other and keep on dividing. This one becomes a baby, and this one becomes a baby. The other one is once it gets to the inner cell mass portion, the inner cell mass separates into two. Now you have two identical twins. The greatest thing about having an identical twin is you've got a guaranteed kidney donor, heart donor, anything donor, because you have the exact same DNA as your identical twin. And indeed, the first renal transplants that were ever successful in the history of medicine were done from one identical twin to the other to overcome the problems of rejection, which weren't understood at all back in those days. About 1 out of 90 kids is a twin, 1 out of 89 squared is a triplet, and 1 out of 89 to the third power, which is 1 out of every 704,000 births, somewhere along in there, is a quad. Most all of the triplets, quads, and twins you hear about these days are caused by in vitro fertilization or by using hormones to make ladies throw out more than one egg. If they're having troubles getting pregnant, you give them hormones to make those curves on the cycle that we drew bigger and better to make sure that they actually ovulate. This is what your karyotype looks like. When I had my two kids, my later two kids, we had amniocentesis done on my wife in both cases. About 12 weeks of pregnancy, she went in, had a needle put in her amniotic fluid. They suck a little fluid out. They look at the cells. They create a picture like this. Remember that a karyotype is nothing but a photograph. It's a photograph. You smear out the chromosomes. You cut each one of them out, and then you line them up and put them in. We have 22 pairs of autosomes, which carry all of our genetic information, basically. And then the 23rd pair is the sex chromosome. If it's an X and a Y, it's going to be a boy. If it's an X and an X, it's going to be a girl. And we did that because we did not feel at that stage of our life that we were capable of raising a baby with bad genetic problems. And she was older. The risk of Down syndrome goes up really high, and we just wanted to make sure that wasn't the case. So my kids to this day carry around their human karyotypes saying, see, I'm not, I'm normal. Look at me. And they carried them to first grade, too. Last couple slides, I know I've kept it a little bit late. The Human Genome Project, we thought it would take 50 years for us to figure out the sequence of everybody's DNA, because each gene is tens of thousands of triplets of DNA and all those A's, C's, T's, G's, and all that jazz. But now with computers and supercomputers, you've heard on TV, one guy, I heard him say, it used to take us 2 weeks to do a genomic testing now we can do 100 in a day. I mean, that's just kind of the way it's gone from the computer use of this. And as a result of that, we now know just about all the diseases that are known to man. We can tell on which chromosome the problems are. And, you know, we know that PKU occurs on chromosome number 12. We know that ovarian cancer, the genes that give some ladies ovarian cancer at such a high rate, occur on number 9. We know that the BRCA2 gene, which causes breast cancer and ovarian cancer, like Angelina Jolie has, which would have given her about a 75% chance of getting breast cancer and another 60% chance of getting ovarian cancer. She said, hell, take them off. And they did. And they took her ovaries out as well. So she's not going to get those two kinds of cancers, but she was at an extremely high rate. And so that's the first kind of information this has been really helpful for. It's really helpful to know if you're a carrier for a bad disease, if you're thinking about having children but you're not really sure you want to, or you're not sure you really want to marry that guy because he's got a gene that's not so good in his family and so forth. Obviously, the next step in all of this is they're starting to clip out genes and either take them out or replace them with normal genes to solve genetic problems. Yeah.
So when my husband and I had kids, or had our first, we sat there and we talked about, you know, what's your family history, what's my family history. We did all of that. They did a preliminary screening and they found out that I'm a carrier for cystic fibrosis.
Yeah.
Which
That
we
deal.
had no idea of.
Mm-hmm.
Thankfully he's not, but it was really eye-opening.
So half of your kids are now carriers, probably.
Yeah.
The odds would be that half of your kids would be carriers. And the reason I gave you that exercise is on the